And so I want to welcome everyone to Humboldt State. This is a week event, this entire week. This is our Canadian event. This is our Canadian way to celebrate Humboldt's contribution to being laying out the foundation for the field of biogeography. And the International Biogeography Society has centered efforts in organizing events such as this one that we have across the world. I've sat in one in the morning today. And so please visit the IBS webpage where you can find more information. There are many coming events throughout the week, so that would be great. So we have today an excellent lineup of talks today that celebrates our Canadian event, but also we want to have a discussion activity. So I wanted this to be a little surprise because we're going to have a few questions that we prepared that will sort of put some interesting perspective into the research challenges in biogeography. And so please stay for that, or if you come later, please stay. So this has been great. Hundreds of people have registered in the past 12 hours, 40 people registered. So this is demonstrating the great interest that people have in biogeography and that they're missing biogeography. Or they maybe just forgot how to register, but I prefer to think that the former is a better explanation. So on this note, I would like to thank first Kate Jones for accepting giving our introduction talk. Kate, in some ways, is our ringer because she's not located in Canada. Everyone else is located in Canada. So I thought that would be best not to pick a Canadian to do this. So Kate is a professor of ecology and biodiversity and the director of Biodiversity Modeling Research Group at University College London. Kate research investigates the interface of ecological and human health using statistical and mathematical modeling to understand the impact of global land use and climate change on ecological and human systems, with particular focus on emerging infectious diseases. And she's going to talk to us about our planet, our health, ecosystem approaches to forecasting zoonotic disease. Thanks, Kate. Thank you very much.
Thanks very much, Pedro, for the introduction and inviting me to give a talk to you all from my spare room. Okay, so I'm going to talk to you about my research, trying to understand how the health of our environment is intimately connected to human health and well-being, with a particular focus on emerging infectious diseases. And those are actually zoonotic diseases, the ones that go from animals to humans, which is obviously, of course, this is of particular relevance now in the current pandemic. So I'm going to try to share my slides. You see that?
It was great before.
It was great before.
No, yeah, perfect.
Okay, all right, so I want to just take you through a whistle-stop tour of some of the work that myself and my group have been doing to try to think about the linkages between the health of our ecosystems and the health of our planet and human health and well-being. Okay, so when we start to think about the links between ecological health or environmental health and human health and well-being, you can see a kind of vast gap between the two kind of sectors, ecology and public health. But actually there's one area which has been quite active over the last few years, and that's the links between public health and climate change. And this started around 2015 with a massive Lancet countdown report, and Lancet is one of the biggest medical journals in the world. And they are, the public health community got really excited about the links between climate change and public health. And this started a hugely long and actually really long engagement with the public health community. And that's been really brilliant. And that's been a really big sea change in our kind of conversations between environmental scientists and the public health community, because it is very siloed. However, the kind of more intimate links between land use change and biodiversity loss and public health are not very well understood, and definitely within the public health sphere, it's not really discussed very much, and the integration is not there. So, okay, well, I'm trying to think about what those are with particular relevance to disease. And, you know, I don't need to tell all of you, but we're disrupting our natural ecosystems, and habitat loss and land use change is one of those key threats to biodiversity of different impacts depending on what group you're looking at. But also that's driven by huge amount of humans, which is human populations, and then our actual growth rates slowing down. But because we've got such inertia in the system, we're looking at around 11 billion people by the end of 2100. And our kind of road densities has been fragmenting our landscapes, so that we are disrupting ecosystems in that way as well, in terms of fragmentation. And of course, pollution is also a really big problem in terms of artificial light, but also other pollutants in our environment. So we are doing massive disruption to our ecosystems, and that of course has impacts on our the kind of things that we get from our ecosystems, termed ecosystem services. I'm sure I don't need to tell this crowd this, but this is one infographic from a 2011 report that kind of assessed the state of ecosystems and ecosystem services in the UK. So this is kind of diagram showing all of air, land, water, and all living things in the green boxes, and they're packed up into ecosystems which are delivering services of some kind. And those goods help our human health and well-being in terms of economic value, health value, and shared societal value. And of course, there's a feedback loop where you have social feedbacks and drivers of change impacting these ecosystems, and therefore the goods and services produced. And this down here is just some examples of those ecosystem services that are produced that are important from our ecosystems. And this isn't just me saying this. This is kind of now implemented in a huge number of different international, local, national, and international documents across the world. So just really local, from the city where I'm in London, we're trying to implement this kind of ecosystem services and natural capital into our accounting of the state of the environment in our city. But also our UK government is putting that into our 25-year plan to improve the environment, which is now, it's now fed its way back into an environment bill, which is going through Parliament at the moment. Well, I don't know what's happening to my Parliament at the moment. It seems to be in disarray, but hopefully it will go through. And then of course it's part of Best, which is our kind of biodiversity and ecosystem services side of the IP, that equivalent to the IPCC, so Intergovernmental Panel. And of course that's also made it down into the Sustainable Development Goals, where they're recognising that 14 and 15 are kind of key to the other Sustainable Development Goals to meet. So as biogeographers, you probably know that already. But I guess what I'm really interested in is this bit down here, which says diseases and pest regulation. So how does losing biodiversity and changing ecosystems change the disease and pest regulation and diseases that might come from us? And that's a really critical issue that I've been devoting quite a lot of my research career to. Like, what are the mechanisms? And then can we go from these mechanisms into prediction, so we don't end up in a situation we are in now where we're all in our houses? So like, how do we go from understanding the fundamentals to then making predictive models about the future? Okay, so if we want to take a kind of ecosystem approach to this and understanding how changing ecosystems are increasing or decreasing the number of zoonotic and diseases transmitted from animals into humans, then we need to have a think about the transmission pathways. And I've just put a couple of infographics here just to show you the kind of massive range of transmission pathways with which viruses and other pathogens get from animals to people. So this is from an infographic from London School of Hygiene and Tropical Medicine. So you can get viral pathogen transfer through air. So the current coronavirus is like that. You can also have vectors which are transmitting those pathogens into people. So, for example, malaria or dengue fever. You can have fecal-oral transfer, or from animal body fluid in cuts like Ebola, for example. Or you can have a mixture of things. So you can have pathogens coming from cattle directly, or you can have a wildlife host and then a kind of domestic host and then into a human, or you could have a vector involved in that process. So I guess what I'm trying to say is that the transmission pathways are quite complex. And what I'm trying to understand is how ecosystem disruption impacts those transmission pathways to then create a situation where you get a transfer from these pathogens into humans. So this is really important because over 2/3 of all human infectious diseases do have an animal source from either domestic species or from wildlife. So it's really important to try to understand and try and predict what animals and what of the transmission pathways are going to be of the highest risk. Okay, so although this is a little bit, the evidence is a little bit mixed and lacking, we think that these events are increasing over time. So this is a paper I did in 2008, which showed a kind of increase over time of these emerging infectious diseases events, which is the first time that a pathogen has transmitted into humans from animals. And there's some other evidence that this is increasing. So, you know, taken as a whole, we think this transfer and these transmission pathways are being changed more and more, and we're getting more of these events occurring. Okay, so of course this is particularly relevant now because of COVID, and COVID-19 is also a zoonotic disease as far as we can tell. The current evidence is suggesting that the closest related virus to the one that's found in humans has been found in a bat. So that's not to say that bats are the host, but that's our top candidate at the moment that we think that there's highest likelihood of coming from a bat, because it's closely related to the ones which we've found in bats already. So we don't exactly know what the transmission pathways are. It could have been a wet market. That's just one point of transfer. It could have been in agricultural areas where this happened. We just don't know at the moment. And we also don't know the other pathways involved. So there could have been a number of other species involved in this pathway too. But at the moment, I love bats, by the way, so I try to deflect blame slightly from the bats. But I guess what I'm trying to say is that the COVID-19 is a zoonotic disease, but it's actually not really relevant to point a finger of blame at different species. It's more about how we as humans are changing these transmission pathways. Okay, so let's have a look at that in more detail. What do I mean by changing transmission pathways? So I told you in that slide before about the variety of transmission pathways that could be operating to get pathogens into humans. But I guess it's more about how we're changing those transmission pathways with anthropogenic activity. So this is a kind of diagram from a paper, a really fundamental paper in the field of trying to understand what's happening, by Peter Daszak in 2000. So you've got these overlapping Venn diagrams here of emerging infectious diseases, or just pathogens in wildlife, domestic animals and humans. And you can get transfer between them by changing these transmission pathways. So if you have a pathogen in a human, to get to other humans, you would have changing global travel, urbanization, or some accident that's happened in the lab. But if you were thinking about human and wildlife, you might be thinking about human encroachment, of changing the kind of rate of exposure of humans to wildlife pathogens, or some kind of translocation of wildlife, or and if you're talking about wildlife and domestic species, you might be talking about domestic and agricultural intensification of domestic animals. So there are many pathways that you could be changing when you're changing anthropogenic pressures on the planet. But okay, that's great. It's really great to have this kind of overview, but how exactly does that work? And how do you then use this understanding to make predictions about what's going to happen next? And that's really been the focus of a lot of people's work over the last 20 years, is going from this kind of overview to actually what's happening or what will happen. So there's a huge debate about how ecosystem change influences the risk of having a pathogen transmitting into a human population. And there's a lot of debate. There's been a lot of debate over the last few years about how exactly that happens. And we published a paper in August which kind of tried to really understand the mechanisms of exactly how this might happen. So what we did was look across all of the experiments all over the world, and that had looked at a different gradient of human pressure. So ones which had looked at low pressure, and then ones which had looked at areas under high pressure in the same experiment, and looked at what species were present in those low pressure areas and ones which were present in the high pressure areas. And then independently, we looked at what types of species they were and whether they were actual hosts of infectious diseases that we can get. So what we found was that land use had a really big impact on the amount and the abundance of pathogens in the species which could survive in those areas. So we have some really good evidence across the planet that when you have a human-dominated system, species which can survive in these human-dominated ecosystems, so passerine birds, rodents, sunbats, not all, they're the ones which can actually have more of these species, pathogens which can infect us. And so therefore they're a bit more, we're creating these risky landscapes by changing the kind of ecosystems which are then present. So that was a really interesting study in trying to link not just something about biodiversity and ecosystem change to pathogen risk, but actually how land use might be a useful proxy for some of these mechanisms. And so, you know, how land use is changing, is filtering out the species which are present in these kind of more risky landscapes. And we were seeing a really big effect in urban areas, which is super scary. Although I would say that the sample sizes for urban areas are quite low in this study, just because people don't focus on them very much when we're looking for these types of experiments. Okay, so we've been thinking about this for a long time now, and we tried to think about going from those kind of mechanisms to prediction. And one of the frameworks we think is really, really useful is a kind of a hazard exposure vulnerability model. And this is used in climate change as well, climate change science. And where you have risk as a kind of intersection of the hazard, which we're saying is the kind of zoonotic hazard, so the underlying ecological systems which are in place. And then your exposure is how you then interact with those land uses. And then you have to factor in vulnerability. So how much do you have access to healthcare, or how does conflict change that access to healthcare? Or what's the underlying susceptibility or immunity of the human population? So I guess what we're trying to put together here is the ecology, but also the human epidemiology, and then socioeconomic change, all in one kind of model, which is actually quite tricky. So a lot of, just one dig at the epidemiologists, the public health people, a lot of public health people just focus here on the exposure and the vulnerability to work out risk and miss out this kind of ecological hazard completely. So, but unless you actually put it all together, you're not going to get your predictions right. And I don't think we have been getting our predictions right in the past. Okay, so trying to go from that into something that you can actually model has been tricky. And this is something that we've put together to think about it in a bit more detail. So you've got all the yellow bit here is the physical environment. You've got the host that's, hosting this wildlife or domestic species, which is hosting the pathogen here in the greener box. And then a subset of that is actually infected. Those are the hosts that are infected with the pathogen. And then you have this white box here, which is the human population, and they're moving about. And then you've got this very dangerous spillover zone here, where you've got pathogen spillover into kind of infected populations, and humans can move about in in their populations across the planet. And then every single one of these boxes is then impacted by some socioeconomic drivers. So it's incredibly difficult to model if you're just doing it in a linear way. And I guess what we're trying to say here is that you need to do it in a kind of systems dynamic way. So you need to understand how socioeconomic change is impacting every single one of these boxes to then figure out how your transmission pathways are changing. Okay, so that sounds incredibly complicated, and I'm just going to show a couple of examples of the work that we've been doing. So we first started thinking about this with Lasso Fever. So Lasso Fever is hemorrhagic fever. It's spread by these lovely little cute rats here, Mastitis nataliensis. It's got a huge distribution. It's an agricultural pest. The pathogen is spread through feces and urine, usually in people's homes or when they're collecting their crops in. So it's a proper disease. It's got its own book, which is a good sign, and it's got a really interesting distribution. In fact, it's a really big puzzle. So it's just found in West Africa, and that's quite weird, given that the fact that Mastitis is sub-Saharan widespread agricultural pest. There's some evidence that the numbers of Lasso fever cases are increasing over the years, and it's very, very understudied. So we could perhaps be seeing about 300 cases a year. It's got a lower mortality rate than Ebola, but that would make it the kind of a hemorrhagic fever that's infecting more people than Ebola ever has, which is really quite scary. So we just tried to think about, could we explain firstly the kind of zoonotic hazard that the disease might have, and people hadn't really been looking in this way at this disease. They've been kind of looking at the number of cases in West Africa rather than trying to understand the links to the agricultural pest. So we did a whole GenBank sequence analysis, and we collected some more samples from Sierra Leone and Nigeria, and we figured out that there was actually 3 clades of this species, at least 3, and the Lasso fever has evolved in this clade on the left-hand side here, and the split from Lasso fever from its closely related arenavirus sister taxa happened later than the split that this clade here split off for the rest of the Mastitis. So it was evolved in this clade, and whether it can jump to others is another question. And this is quite important to know, because if it can make that jump across these clades, then you would have an infected rest of Africa. Okay, so we've kind of tried to pin down the zoonotic hazard in some way, in that's why there's a restricted distribution. So we wanted to try to understand what the cases were telling us, and whether we could make a predictive model based on the cases in West Africa. And a very typical approach for many biogeographers and many ecologists is to do a correlative model. So you have your field observations, your environmental layers, and you do some kind of machine learning or model fitting to your spatial layers of environment and human density, etc. And you would come up with spatial predictions for the areas you haven't sampled. So this is a kind of standard approach in ecology. So could we use that to understand what the ecological hazard might be, and then try to understand the case distribution? So this is our first attempt at doing this, where the greener the values in the map are the riskier parts of our model distribution. So, the things that have gone into it that are really important are the host occurrence. So we modelled that separately. So where we've got areas which are suitable for this rat, you've also got a high likelihood of having one of these outbreaks. And also human population density as well was important, as well as rainfall and the amount of kind of crop yield that was in those agricultural areas. So this is a really standard approach, and yet I think it's it poorly characterized the data because it's completely biased, and it's got a poor predictive power, because when you change the environment, it's something that this model hasn't seen, and it does poorly in future scenarios. So going from understanding these kind of mechanisms to prediction is very tricky with these correlative approaches. So, and also there's a massive hospital here, that's the only hospital in that region to diagnose Lasso Fever, and similarly in Nigeria that's the case. So we were very suspicious about this model. So what we tried to do was go put some more mechanism into this, so we could try to mash up a kind of traditional epidemiological model with an understanding of these kind of ecological and environmental processes. So this is the kind of model that we came up with in the end, after a lot of head scratching, was to kind of take the approach of many epidemiological models where they compartmentalize human populations into these compartments of whether they've had the disease, or whether they're infectious, or whether they've been removed from the population, recovered, or have died. So we cut up this whole area into these grids, 1 kilometre grid squares, and then we had human populations in these compartments. So you're either susceptible or infectious. So Lasso Fever only goes from animal to human, and it doesn't go from human to human. So we use this kind of modelling approach, stochastic modelling approach, but then informed the models with real data from human population density from satellites, but also using our distribution model of the suitable landscapes for the rodent, and a few kind of assumptions in the model, which I won't go into here. But then we've got a kind of ground up model of what we think is the kind of risk, going from the hazard and susceptibility of the humans and the exposure. So this is our model, which is a much poorer fit to the data, but we think is probably a better understanding of what the mechanisms are, and therefore what the prediction of what the cases should look like. So we did this in 2016, and then we got talking to the Centers for Disease Control in Nigeria, and they've been setting up these the first amazing systematic surveillance of Lasso Fever anywhere in the world, and they've been doing it over the last few years really seriously, and have been doing it a little bit since 2012. So we've been collaborating with the fantastic team over there, and some of our colleagues, other medical colleagues at UCL as well. So our kind of approach, where I took this kind of statistical approach and then statistical mashup mechanistic approach, we compared that to the actual data that they've been collecting over the last few years, and it's a much better fit. Our statistical mechanistic approach is a much better fit to the one that they've been coming up with in their surveillance. In fact, we've been using their data and our approach to make much finer grained predictions across space and time. So with our kind of approach, we've been able to pin down some of the temporal sequence associations with the cases, and we can predict quite successfully 3 or 4 months ahead of time where and when Lassa fever cases will spike, which of course is incredibly interesting for the Nigerian CDC. And I think it's a really interesting way of trying to mash up these mechanistic and correlative approaches to understand the impact of how environmental change and human changes are impacting the likelihood of spillover. Okay, so when we were doing Lassa fever in Sierra Leone and collecting data in 2014, the Ebola outbreak happened, and our team was out there at that time. And so we started to think about this in terms of not just Lassa fever, but maybe we could apply it to other diseases as well. And Ebola is incredibly horrendous disease, but it's incredibly interesting as well in the fact that it also goes human to human. So the model, the kind of compartmental model, has to be a lot more complicated. So here you can see the susceptible compartment, then you have an exposed compartment, infectious, and then recovered, or you'll get removed, i.e. you're dead. But because Ebola is so horrendous, you're still infectious 2 days after you're dead. So we have a special funeral compartment here in the model. So this, we cut up all of Africa into, or the planet, but I'm just showing you Africa here, into 1 kilometre grid squares. And we have this stochastic model operating within this grid square, and then exposed and infectious people can move across grid squares along existing transport networks. And then what we can then do is understand the kind of zoonotic force of infection from here by mapping out the kind of occurrence of the probable hosts, and then understanding how many humans are in each grid square, and then stochastically letting it run over thousands and thousands of repeats, replicates, to create these landscapes of risk. So it's trying to parameterize this exposure, hazard, and susceptibility model within this framework. So it's quite cool because you can, play with the present day conditions and then change them into different future ones. So you can get an understanding of how global changes in different scenarios might impact your risk. So this is the result. So because this is a stochastic model based on things that we know about how Ebola progresses through these compartments, and then parameterized by understanding where the hosts are and the kind of risk from those in that transmission pathway, it doesn't have any outbreak data in it. So it's kind of from the ground up. So what I'm showing you here is a risk map. So yellow bits are more risky, of Ebola spillover and transmission. And I've overlaid the actual number of where the outbreaks have been historically. So you can kind of see how our prediction matches the outbreaks that we've seen. But these outbreaks and where these outbreaks were never went into the model. So this one shows your spillover and then subsequent spread. And this one here is just spillovers. And this one is then areas which would show, if something happened, they would show a huge amount of cases. So this is like catastrophic number of cases. So one of the things I'd like to point out is these asterisks here. So when we did the modelling, this was early 2017, we published it in bioRxiv, and then the new outbreaks in DRC happened, and these were really well matched to where we said that if an outbreak did happen, the kind of susceptibility of the human populations is so high that these would cause massive outbreaks, and they have. And so we predicted these outbreaks way before they actually happened. So that's just could be just a nice story, but I think that this approach has got value in terms of having a set of fundamental principles about how the pathways are changing, what the pathways are, how the pathways change, and then you can then play with future scenarios to understand how the risk changes. So different scenarios could be different RCP scenarios of different greenhouse gas concentrations, or different different shared socioeconomic pathways as well. So you could kind of mix those up to understand how that's going to impact on disease emergence. So maybe it shouldn't take the actual numbers very seriously, but I think it's a really interesting framework to go from trying to understand these mechanisms to forecasting. Okay, so conclusions then. You know, in order to put the ecology bit into predictive models, we need to know much more about how reservoir hosts respond to global change, and how these ecosystems are changing in terms of the hosts present. You need to understand and model zoonotic disease emergence with a systems dynamic approach, because it's a very complex system. But I think there are exciting new developments between stochastic modelling, mathematical modelling, and correlative environmental modelling to come, and I think that's really cool. And finally, we definitely need much more ecological thinking to understand human health and human futures. Thanks very much.
Thank you, Kate. Please, let's use our little virtual hands to, thanks, Kate. Thank you. I have, because you came from so far, I'm gonna ask
so far.
I know, very far. I'm gonna ask you a few questions. First question I have here, in the clade diagram, none of the West African points were actually located in Nigeria.
Yeah.
Do we know the Nigerian cases belong to that clade and not the clade which has members in Niger and Cameroon?
We do not, and I think that would be a brilliant thing to go and find out. So it's just very difficult to sample in that area. So I think it would be a really, really good thing to have a look at.
All right. How much does
the northern bit where all the Boko Haram terrorists are?
How does the activities, behavior of people matter in the transmission of these diseases? Could that be factored into the models?
Yes. So in the Ebola one that I showed you, we actually had R0, which everybody knows about now, changing over the course of the year that we modelled. So we had 20,000 repetitions of that year. But then as people were infected, R0 goes down. But also as you put more effort into controlling the disease, R0 goes down. So you can change the R0 as you kind of increase or decrease poverty or decrease conflict. So we tried a few things there to you know, realistically decline R0 over a course of an epidemic. So for example, if you got Ebola in London, you'd be whisked off to the Royal Free, where we put all the Ebola people that got infected with Ebola, and so it didn't spread across the whole network. And you know, that kind of thing we tried to capture with increased healthcare provision. But if that happened in Freetown and Sierra Leone, that would be kind of different. So there are a number of ways to capture that, but we tried to think about that.
All right, thanks, Kate. Well, I have other questions I'm going to send to you, and perhaps you're entertained to answer them. Just for the sake of time, we got Jan Sunday is going to be hosting the mini talks. So I'll pass the baton to Jen. I'll you know, you do your own coughing. So
baton received. Okay, yes.
Exactly.
Everyone knows if you hear a strange cough on the through your speakers, that will be our 9-minute mark, or 1 minute more to go for all of our next 10-minute speakers. And so we're a little, I'll keep everyone to 10 minutes from now, so we'll sort of be a little behind in the schedule. But without further ado, I will introduce Carissa Brown from Memorial University, who will be talking to us about biotic constraints on tree range expansion. So over to you.
All right, and I will nervously await your cough. Okay, so the beauty of being part of the International Biogeography Society is that you get to quickly go from Africa to the boreal forest in less than 5 minutes. So I'm going to talk to you about biotic constraints on treeline range expansion. And I'm talking to you today from the traditional lands of the Biotic and Mi'kmaq people on the island of Newfoundland. So just to start us off, I want to start with a little bit of context of ecological inertia. And if you recall or haven't learned before, it is an ecosystem's ability to resist change when stressed. So all the biogeography research that we're talking about right now is under the context of global change, climate change, and changes in environmental conditions. So at a species range edge, like the northern edge of the boreal forest, where it transitions to tundra, we would expect as environmental constraints on tree distributions lessen, that seeds or propagules dispersing beyond the range edge would germinate and establish, mature to reproductive maturity, and sustain a population, expanding that species distribution. But we also know that that seed is not dispersing into an empty landscape. It's dispersing into an area that's already occupied by an intact community. And so potentially the substrate or ground cover is different than within that tree population's historic distribution. Perhaps there's species occupying the microsites that the seed would do best germinating and establishing in. There are other species present in that system that the tree population is potentially expanding into that might have positive or negative effects on that species' success, like predation or disease. So that seed, those propagules dispersing into new habitat might encounter this, this ecological inertia if that system, which if you think about it is the southern edge of its distribution, if it's able to withstand the stress of changing environmental conditions, it might be able to resist the establishment of new species dispersing into it. So how do we tease apart these kind of complicated questions related to abiotic and biotic constraints on species distributions? Well, in our research group, we do manipulative field experiments at range edges. So hold on to your seats. We do that at the northern edge of the boreal forest where it transitions to tundra, but also at the southern edge where we expect temperate tree species will expand into currently boreal forest stands. And one of the methods that we've been using for about the past 7 or 8 years is predator exclusion as a means to tease apart the relative effects of abiotic conditions and biotic constraints. So to put it the most simply, you put a cage on it. Actually, you put a cage on half of it. So whatever experimental treatments and manipulations we have also happening in our field studies, we will often put a cage on half of those treatments. So perhaps there's different substrates that we're testing range expansion questions. Perhaps there's burned and unburned treatments. We apply seeds to all of our treatments. And then the next field season or several field seasons in a row, we'll come back and see where seeds remain or where seedlings have been able to emerge. And this lets us test uncover the relative effects of these different factors controlling a species' ability to establish. We use a number of different kinds of exclosures depending on the questions that we're asking and the landscapes that we're working in. So we have found biotic constraints on tree range expansion in a number of systems around the globe. And so I'm going to give you some evidence from 3 or 4 perhaps studies. So first, and I'm going to do them kind of chronologically over the development of this part of my research program. So let's start in the boreal temperate ecotone, looking at sugar maple distribution along Alpine slopes in southern Quebec. And this is work that I did with Mark Felland at the University de Sherbrooke. And so we planted a field experiment across sugar maple's distribution from within sugar maple forest near 100% sugar maple cover, across the edge of its distribution and beyond into balsam fir forest. After the first year of field experiments asking other questions, we noticed that seed predation seemed like it might be playing a pretty big role at the range edge. So in the fall, we put out cages over half of our seeded plots, left the other half unprotected, and came back the next spring. So we're looking at number of intact seeds on the y-axis, and there's a maple seed on your screen there. On my screen, at least, it's about life size. So they're nice big chunks of protein and carbohydrates for seed predators. Within sugar maple's distribution and at the edge of its distribution, if we put a cage over our seeds, all 5 seeds we planted remain. If we don't put a cage, we lose 1 or 2 seeds, presumably to predation. Of course, we don't have evidence of direct predation, so it's presumed predation. Same at the edge. But if we put a cage on our 5 seeds beyond sugar maple's distribution, beyond its upper elevational range, we get 5 seeds if there's a cage on it. If there's no cage, no seeds. So this is a range edge that's quite heavily controlled by seed predation, by seed predators. Moving to another alpine tree line, but this time, or an alpine tree distribution, but this time in the Swiss Alps and with coniferous species. This work was led by Esther Fry and is part of the global tree line range expansion experiment that I lead. Again, we see these sort of differential effects of predation across the tree species range. So as we move from forest to tree line to untreed alpine or alpine meadows, now we're looking at emergence, so beyond these seeds have already germinated and established. In the forest, it doesn't matter if you have a cage or not. Actually, you're not going to germinate anyway. There's other things going on there. At the tree line, a cage results in more, or under cages, we have more seedlings present. And at alpine tundra or alpine meadows, if there's a cage on it, we even have a bigger effect of protection from predators. Moving on to the island of Newfoundland, where I'm located right now, our forests here are predominantly boreal forests, but we do have pockets of temperate tree populations, and we would expect those pockets, those temperate trees, to expand their range under ongoing climate change. So this is work that Piers Evans did for his master's thesis here on the island, and Anna Crofts in the background assisted a lot with it. And again, put cages on things, had lots of background treatments going on. But where we put cages over sugar maple seed here on the island as well, we found that we only had emergence well protected from seed predators, but that was a species-specific response. And so for other large seeded temperate species that have different life history attributes, the cage was less important. This is an observation that we see not only with vertebrate seed predators, but also inverts. So Newfoundland is an interesting and bizarre place filled with many non-native invasive species. And slugs, the redback voles that are eating our maple seeds are one, slugs are another, as well as many insect inverts. And so we've seen invert as part of her master's research. Anna Crofts observed that one of the reasons potentially that no seedlings emerged in her seeding experiment was that they were all being predated upon by inverts, and there's one of the seeds that she observed. So we really wanted to tease apart the vertebrate versus invertebrate seed predation. And so she put out a field experiment across tree line in an alpine slope here on the island, where again, half of the sites were covered with a cage that excludes vertebrates and the other half weren't, but her seeds were planted in these very discrete PVC pipes where we could keep track of every single seed very carefully. And we did that in within the forest and up at tree line, and with an additional treatment of disturbing the substrate in half of the sites. So now we're switching our y-axis. So this time we're looking at predation pressure. So basically big bars are bad. Within the forest, and we're looking at the black spruce population, there was also larcher tamarack there. Within the forest, if you had no cage on you, regardless of whether we disturbed the ground or not, if you had no cage on you, all the seeds were eaten. Now, we removed that vertebrate predation pressure by putting cages on half of the plots. If you had a cage on you, so now vertebrates can't eat the seed. I'll stop saying you are not getting eaten, the seed are. Anywhere from a third to half of the seed was removed. Oh, I heard my cough. I'm almost done. At tree line, so the population range edge, if you had no cage, you were nearly all eaten. And there's a really neat interaction here that if you did have a cage, so vertebrate predation is removed, scarified ground had about 2/3 of the seeds predated upon, whereas intact lichen cover didn't. And so we think that was more of an access to, or the probability of invert intersecting a seed is much higher if we smooth out that substrate. So widespread climate change means that that initial strong environmental filter of species distributions has lessened. And so the next question then is, how will biotic interactions mediate species responses to climate change? And hopefully I've presented you some convincing evidence here that those biotic constraints, those interactions are really key for some species expanding their ranges. Thank you.
Thank you so much, Carissa. So there are a couple of great questions in the chat, but I think we should move on to try to keep on time. So if you can have a look at those, or I can bounce them to you. If you can answer them in the chat, that'd be great. And very next up, we have Laura Pollock from McGill University, who's going to be talking to us about biogeography of predator-prey interactions. Staying on topic.
Thanks, Jen. I'll try. Actually, I like to spend my time just asking questions to the previous speakers.
Yeah.
No, seriously though, I would like to,
at the end, everyone.
Yeah. So I would like to today begin with a story, and this is a book called Owls, and it's written by Herbert Zim and illustrated by, no, I won't actually read this, but so this book appeared last week in our growing eclectic kids book collection, and this book was written in 1950. And in it, it tells the story of, in 1900, so 50 years prior, how everyone thought that owls primarily ate chickens. And so they wanted to basically exterminate, all the owls. And so the U.S. Fish and Wildlife Service, they went out, and as you do in 1900, they hunted huge amounts of owls and then looked at the gut contents and documented in a lot of detail what we know now is the incredibly diverse diet of owls. They eat basically everything, insects, lizards, field mice. One unfortunate owl even tried to eat a porcupine, which obviously didn't end well. But also they showed the importance of field mice in the diet of owls, and they even calculated how many field mice can one owl eat in a year. So obviously this study was important for, public perception of our wildlife, but also it was a really, really detailed study, and this is 120 years ago. And although of course we've got many, many studies now of, what predators eat, we don't actually have as many as you might think. And anyone who's ever tried to assemble this information will tell you that there just aren't that many really detailed studies out there. And so that's what I want to talk about today. What I see is a really, really big gap, and particularly when you want to look at those interactions across space. There just aren't enough studies to do it. Let me just share my screen here. Can everyone see that? I can't see any of you anyway. I'll just assume that someone tell me if you can't. And so what about this, biogeography of predator-prey interactions? Historically predator-prey interactions were considered more in particular ecosystems with species that we knew a lot about. And if you think about them and compared to the other biodiversity shortfalls, I think that there's just not as much evidence or not as much information to go on. Of course, the Linnaean shortfall. So how many taxa do we have named? Do we know that exist? We have over 1 million species named of the 9 million plus total species on the planet. So we still have a long way to go, but we've made a lot of progress as well. The Darwinian shortfall. So this is, how what phylogenetic information is known for the different species. And so we have, tens of thousands of species for this that are known, or at least have some kind of sequence, documented in Germain, for example. Runkian, I never know how to pronounce that, shortfall. This is knowing the functional attributes of species. And, you know, for this, again, we have, if you go in with these big trait databases, we have tens of thousands of species, if not more, documented, at least some knowledge about their traits. Wallacean, obviously this is very important to biogeographers. And, you know, we have at least tens of thousands of species where we know something about their distribution. We might not know a lot about their distribution or know it very well, but still, again, we're really rapidly filling this gap. And so, you know, for example, GBIF now has over 1 billion records for taxa, which is a huge amount. And so this, you know, this data combined with some sophisticated models, and we're starting to get a better estimate of the distribution of species. Again, there's still work to be done. But the Carsonian shortfall, the knowledge of how species interact in their environment and how energy and nutrients flow through an ecosystem. I renamed this one. Don't worry, Elton won't mind too much, hopefully. So there are relatively few studies on this. And this is, if we go into this one, this is an example. So this is a study by Tim Poiseau, and they have gathered a lot of all the studies that they can find, and these studies are hosted on the McGill database. And this is an example of all of these studies. So this is 1,300 of them, and what type of species interaction study they are, if they're parasitism, mutualism, predation. And so you can see that not only are there not that many when you're trying to do something like understand the interactions for lots of species, but they're also really spatially clustered. And so basically in North America and Europe, there are a few studies on predator-prey interactions, but not really globally. There are definitely big gaps. And also even within North America, if you zoom in, you know, to Quebec, for example, which we've done, there really aren't many species at all. And so one of the projects that I've been working on for the last few years is trying to address this gap and doing it from a big picture scale, doing it from the idea of let's try to take a lot of species and infer at least something about them, and then try to make some, you know, estimates of how these interactions vary across the landscape, for example. And so this is the meta-web of all vertebrate species in Europe, so all mammals, birds, amphibians, and reptiles, so about 1,082 species, I think, total, and about 70,000 predator-prey interactions. So an interaction is just when one predator eats one prey. So it's a huge database, and this database was put together by a combination of searching for all of those diet studies in the literature, and then where they don't exist, using experts, bringing in experts to help refine those estimates of who eats whom, essentially. And so this was done painstakingly by some of my colleagues, Luigi Mariano, Francesco Fisitola, and Wilfred Tuya. And I believe the data paper has just come out. I'm not exactly sure. And this is actually not the full MetaWeb. This graphic is a condensed version of the MetaWeb, where all of these groups, all of these animals are grouped into different functional groups. And so there, I think, I forget exactly how many, 30 or 40 functional groups. And so you can start to kind of predict what species are in which trophic level based on some of their functional attributes. But the nice thing, because we have data for every single species, we also have the distributions, the spatial distributions for all of the species as well, because it's Europe and we have tons of data for Europe. And so you can combine, you have this meta-web of the relationships between all of the species, then you have the distributions. And if you match those two things up, you can actually extract from every cell across Europe, you can extract which species are there. And then based on that, you can impose this local web, or of these species, which one of them are likely interacting in a predator-prey relationship. And of course, these are potential, right? We don't know that these species actually interact. We don't know if one actually eats the other, but it probably could if it encountered the other one, just based on the database. And so we've been using this for to ask a variety of questions, which are kind of, classic questions, but we've never really had a good data set to address them. And the first is, what is the effect of one species on another? And so for this, we used species as predators, and we analyzed what is the impact of if you have a prey in that cell, or if you have a competitor in that cell. And competitor is just based on whether or not you share the same prey. And we put in all of the environmental variables that you would normally put in like a species distribution model. So we can basically control for those. So is a species there based on the climate, right? And then is there an interaction? Is that influence above and beyond the effect of the climate? And the answer is yes. So these other species have a huge impact, statistically anyway, on the distribution of target species. And so this is birds. You can see here the birds, mammals. That's a wombat. That's not found in Europe. This is an early graph. Amphibians, reptiles, and so you can see this is the effect of the prey on the predator, on each predator. And so the prey always has, not always, but in many cases has a strong positive impact. The competitors either have a positive or negative. It tends towards negative for some groups. Okay. And then I'll just go through these pretty pictures now because I'm out of time. But you can also do things like map connectance. You can look at species richness and trophic richness. So species richness increases with trophic diversity. Of course, we know that, but it does it more so if you're in a really cold area. So it fills those trophic niches more quickly. And then we can make other pretty maps about the biogeography of predator-prey interactions. So I'll stop there. Sorry.
Awesome, beautiful. Thanks, Laura. Love those. We have some other great questions for you in the chat. But just to keep on time, I'm going to move on. So please have a look at those questions, and then maybe we'll have some more time for discussion at the end. Very next up, we have Gabrielle Muñoz from Concordia University, who will be talking about the biogeography of palm seed dispersal interactions. Go ahead.
Hello, hi. Thank you for the presentation. I'm gonna share my screen. Give me a second, please. Oh. There you go, you can see it, right? Let me set up the timer. Okay, all right. Well, hi everyone. Welcome and thank you for being here. My name is Gabriel Muñoz, as I said before, and I'm gonna talk today about the biogeography of palm seed dispersal interactions. So eating fruits is something that many animals do, and this mutualistic interaction, specifically frugivory, which is an interaction between plants and animals, evolved as an adaptation for the seed dispersal of plants. And it is in this context of dispersal that frugivory becomes an important ecological process because it regulates the flow of genetic material among plant populations. It influences the plant and animal meta-community dynamics and supports a large food web of organisms. So for all these reasons, frugivory has a key role to support ecosystem functioning and regeneration. And this is particularly true for the tropics, where up to 90% of the species plant species, can rely on animals for dispersing its seeds. In addition, over evolutionary timescales, frugivory also has an influence on the co-evolutionary dynamics between the evolution of plant and animal traits. So it's because of this that species traits can influence plant-animal interactions, such as frugivory or seed dispersal interactions. So for instance, if we were to match the functional distribution of plants around the globe with the animals based on their interactions, we could expect to see some patterns such as this, where the upper three panels show the effect of a positive association between plant and animals based on their traits. So for instance, that could mean that larger animals tend to eat larger fruits, for example. The strength of this association will be driven by how narrow or large are the average interaction niches of plants and animals. But we can also find some other type of associations, such as negative, no associations between plant and animal traits, which imply that maybe the population sizes what driven more interactions, and some sort of other like nonlinear correlations, which like over large scales would imply, for example, that some plants or some species of animals have different preferences of feeding preferences at different portions of their ranges, for example. So in the tropics, palms are keystone species, sorry. And palms also cover a wide array in the spectrum of plant food morphologies, from being very small to being very large, such as coconuts, and also having like a wide diverse array of animal seed dispersers. Interestingly, there are like contrasting evolution. There are some contrasting evolutionary histories between major regions for palms and their seed dispersal or animal consumers. For instance, the fruit size of palms are generally larger in the Afrotropics compared to the Neotropics. But in the Neotropics, palms are much more diverse. In contrast, like the consumers of palms like tend to be as well larger in the Afrotropics, but like different extinction pattern occur where like in the Neotropics, like after the Pleistocene extinctions, most of the megafauna went extinct. So these large-scale evolutionary histories between regions lead us to the question: these like evolutionary histories has influenced the large-scale trade matching patterns we can observe for the metawebs of the Neotropics and the Afrotropics? So to answer this question, we recently published a paper along with Daniel Kisling and Christian Troelsgaard in the journal Biogeography, where we synthesize a lot of information that we screened from like over 2,000 articles where we search for pairwise interaction data on the dispersal of palms, basically which animals consume which palms and the dispersal effectively. And with that information, we separated for the Neotropics and the Afrotropics and built metawebs, so aggregated interaction networks. And using this interaction data, we joined with available trade data to search for these trade matching relationships, specifically for size-related traits. We were like frugivore body size and palm fruit length. We predicted that despite these evolutionary histories, these different evolutionary histories between regions, we'll expect to see like the same sort of positive association between palm and frugivore traits, perhaps in different ranges of the spectra, but like they still have both, since there's some morphological limitation of fruit consumption, we'll expect to see like the similar positive relationships. So here is the depiction of the metaweb that we could compile for the Neotropics and the Afrotropics. Here is, this is metaweb is gathered at the species level. So each node here represents a species color coded by the clade, and like a link between them represents an interaction. And the size is scaled to the relative size of fruit length or like body mass. So visually, we can already see some interesting associations between palms and mammals for the Neotropics, for example, but really we cannot tell much about the differences just visually about the difference between these two metawebs. So in terms of taxonomic differences of interactions, for example, we can already say that the Neotropic network is much more diverse, at least taxonomically. So in here, in the x-axis, we have like accumulation curves where like in the x-axis are the species of palms or frugivores, and the y-axis are the accumulated number of interactions that the species have. So we can see that like Neotropics are both like palms and frugivore in the Neotropics are more diverse, have more diverse array of interactions than the Afrotropics. But also palms are more generalist than frugivores in both regions, but palms are much more generalist in the Afrotropics, in the Neotropics, sorry. As we can see here, these differences are much larger. So contrary to our initial prediction, we didn't find like both a strong positive trade matching relationship in both regions. So in here, in the x-axis, we can observe like a scale for the world, like variance of the distribution of world distribution of body mass, average fruit body mass, and all the world distribution of average fruit palm length. We can observe that like for the Afrotropics, there are a strong relationship here. Like the points represents the amount of times that we found a given interaction. And we can explain the absence of a relationship for the neotropics for this process of megafaunal extinctions as I talked before. So we can as we can see, like the megafauna has become extinct in neotropics. So, and we observed that many of these interactions that probably were before, like these larger fruits that were probably evolved in initial to be dispersed by large megafauna, were now like handled by like rodents that has the ability to handle fruit. So a bit, in order to finalize with what I would like to go further with this, we already have collected the trade relationships between like major regions. So we can already using downscaling these networks to using like distributional information and this trade matching relationship, we can already downscale networks by modeling missing links. For instance, here we can also do that depending on like the difference on the functional distribution of like this trophic, the functional distribution between trophic communities of palm and animals. For instance, here we can see the spatial patterns of difference between like the mean traits of palm and animal frugivores coexisting in a given cell in the neotropics. So with this information, we can already start asking questions further that not just are about the biogeography of interactions, but also about the biogeography of interaction niches and network assembly processes. We can also perhaps link, like, for example, where like how effects of what will be the effects of climate change on sea dispersal of ponds, particularly, for example, that with the future climate change projections will modify functional diversity of these communities, that will in turn modify the network assembly processes and interaction niches. But finally, a big challenge we need to do is like keep collecting data, because as despite this representing a large effort, there's still many interaction missing, interaction information missing, which we can model, but like we still need some validation. So therefore, like is imperative that we can keep the data collection and keep up with this type of synthesis. Yes, thank you. And so just to finalize, I would like to give a special thanks to all these data contributions, because I spent a lot of time searching this data, but there was a lot of people behind that spent time in the forest trying to collect and publish this data. So the Biomag team in Amsterdam, the Aalborg team in Denmark, and the Community Ecology Biogeography Lab here at Concordia. And with that, I would like to take questions if there's some time. Thank you.
Awesome, thank you. You have sort of 30 seconds. But I do, so I'd actually, Gabriel, I'd like you to, there is a question in the chat that I think you might be able to address from Juan Andres Martinez Lanfranco. But while you're having a look at that, I'd like to pass the baton back over to Pedro, because he's just gonna do a little chat with the whole group.
Yeah, right. So we're always curious to know where people are attending from today. So we're gonna do a quick poll, just basically on continents. And we have lots of participants right now, so it seems to be a good time to do it. I'm gonna enter the poll now. So not surprising, lots of people here in North America, and we're kind of spread out all over the world. So thanks for that. All right, so that gives me the chance to introduce our next speaker, who is my co-host here today, Jen Sunday from McGill University, and she's going to be talking about what range shifts can tell us about species interactions. Thanks, Jen.
Awesome. Thanks, Pedro. Thanks, everyone, for attending. It's really great to see everybody in the chat, and I hope we get a chance to discuss at the end. So I am going to talk a little bit about what range shifts, so changes in species distributions, can tell us about species interactions. And so we know that climate change is already happening. We We can make predictions, but we can also look at what's already happened through the past. We know the species range distributions are on the move. They've been happening and they've been attributed to global climate change. And so on this plot, I'm showing the change in the latitudinal distribution of birds in North America from the Audubon Society, and compiled changes in fish latitudes around North America, mainly compiled from Pinsky et al., but also compiled in Ocean Adapt from NOAA. And we know that a lot of these range shifts, they're basically everywhere we look around the world. When we look for range shifts, we see them occurring. So they are occurring globally. And this paper shows just a smattering of range shift studies from around the world, and also sort of showing in which times they're affecting ecosystems, human and cultural, societal systems, and governance systems. Okay, but range shifts are also variable. And here's just some data from a recent paper from Fred Stone Herman showing the poleward, this is the range edge. In A, it's the poleward range edges, and B, it's the warm range edges through time. So this is fairly high resolution data on species range edges through time. So it's great data. Even fish in the same region are not doing the same thing. They've experienced the same temperature change, but they're not responding the same way. Okay, and we know that organisms live in multi-species environments that are probably playing a role in how these range shifts occur. So one of the central tenets of ecology and biogeography has been that climate influences continental range distributions. So a little example, but we've already seen today lots of examples of how range edges are also possibly controlled by biotic factors. So this has been long postulated by great ecological thinkers of previous times, that warm range edges are more influenced by biotic interactions, while cold range edges are more influenced by abiotic interactions. We have a little, and so sort of the classic example of this is Connell's work in the intertidal. This is a very small scale. There's been some more work showing that places where it's easier to measure rather than latitudinal ranges, looking at elevational ranges, showing that at the warm range edge, it looks like competition limits some plant communities, whereas at the cold range edge, abiotic limits them. I have a little bit of work showing that thermal tolerance niche, when you compare thermal tolerance niches to realized geographic distributions, you see, particularly at the warm range edges of terrestrial species, a big gap where they're limited. They seem to be limited by something other than their heat tolerance, but it could be lots of things. And so what I want to put forward is we've had so many range shifts occurring already. If we're taking really good observations of those changes through time, we can use those range shifts to test hypotheses about the mechanisms that limit those ranges. And this isn't just my idea. This was at least put forward by Eugene Odom in 1958, who said that oftentimes a good way to determine which factors are limiting to organisms is to study their distribution and behavior at the edges of their ranges. One or more environmental factors may undergo a sudden drastic change, thus setting up a natural experiment, which is often superior to a laboratory experiment. So we have natural experiments. If we study the responses, we can tease apart the mechanisms at large scales. Okay, and we have examples of range shifts from range shifts where we know species interactions are playing a role. And so a really simple example is in the Tasman Sea off of Eastern Australia, which is a global warming hotspot. We've observed sea urchins. So I'm taking you into the marine environment now. We've observed sea urchin range expansions. And once those urchins expanded their range, we know that they're an ecosystem engineer, and they have decimated kelp forests. And that's affected, of course, a lot of the species that are associated with those kelp forests, all through species interactions. So the range expansion has led to other range contractions. Okay, and so I'm just going to show you some results of a study also in this region where we asked, can ecological traits explain the differences among species in their range shifts? So I hinted that there is lots of variation in species range shifts, and so we wanted to ask, can we take a look at that variation and at least attribute it to certain traits, and through that infer the role of species interactions? It's pretty indirect, and I'll get to that shortfall at the end. But for starters, here's what the climate velocity has looked like in this region. So this is showing the temperature of the coastal air, water through time along this big latitudinal gradient. And so if you follow the little gray lines, you can see a temperature isotherm and how it changes through time, and it's moving to the south. So it looks subtle here, but this is rapid change. This is 4 times the global average of climate change. And as a result, lots of species have, of course, communities have shifted, species have shifted their ranges. So here's what this is compiled from 7 independent latitudinal field study programs. So studies where in the past, the black lines are showing the poleward range edge. So this is the cold range edge of a whole bunch of different marine species, coastal, nearshore marine species. And so most of the time there's just 2 observations. So at one time point, the range edge was determined for a bunch of species and then redetermined at another time point. So it's noisy data. It's based on most of the time on 2 time point observations. But some of the things we can see is that just to illustrate this, a line that declines quickly as a species where we think the range edge has shifted a lot over this time. And a line that shifts not very much is a range edge that we think, or we haven't observed a range shift in that species. So there's a lot of variation, but on average, the mean shift is entirely in the direction that's predicted by the change in temperatures and not far off in terms of the amount of change. Okay, but can we use ecological traits to explain some of the variation in how quickly those species have responded? So first, species have experienced different amounts of climate change. So I'm just gonna list hypotheses here. So one sort of variable that we think is gonna explain the variation in range shifts is what the velocity of the isotherm movement has been over time for that species. We also hypothesize that traits that have to do with the species' ability to arrive into a new region will be important. So their adult mobility, their larval dispersal ability, and their body size. So these are sort of thinking about the traits of the species. And here's where we get a little bit more into species interactions, which is that the prediction that species that are better able to establish, after they arrive, can they establish in potentially an entirely new community? The temperature's right, but the species are all different. And so we predict the species with a wider diet breadth. We have different hypotheses for what trophic position would do to your ability to change your range. And we also predict that latitudinal range size should improve your ability to establish beyond your previous range. And I'm just going to explain that here. Why do we think that? Well, species with larger ranges, we expect to have larger local abundance, so they should have more arrival, propagule pressure. And we also think that they are more ecologically versatile. They already live in lots of places. Maybe they can live in more. And so why is that doing that? Here we go. So, so this is sort of picture I have in my head. A species with a large latitudinal range might be limited really by temperature. There's a species with a small latitudinal range might be limited by factors other than just temperature. So the lack of a habitat or a strong competitor. This is starting to fall into species interactions especially for marine systems where the main biotic filter will be temperature. Okay, and so here's our prediction. And here's what we find. So I'm totally just summarizing this with a cartoon. So we found that the velocity of the rate of climate change was important. The adult mobility was important. So fish shifted their ranges faster than crawling and sessile adults. The trophic position was sort of a weird finding. We found that omnivores were better at shifting their ranges, possibly because of their great versatility, possibly just because barnacles it was really the barnacles. So there's sort of more to sort out with that. But then also that the species with larger latitudinal ranges indeed shifted their ranges faster. And here's what that looks like. So I've got climate expectation on the x-axis and poleward range shift on the y-axis. And the gray scale is showing, just categorizing the latitudinal range size. And so species with larger latitudinal ranges had a faster response rate, a steeper slope to the climate expectation. So they shifted their ranges closer to what we expected them to do. And these species with these larger ranges looked like this. They were big, they were small. That what they had in common in our models was that they had larger ranges. So possibly, thank you. So getting back to species interaction. So what we can see from this is that we have some evidence here that narrow ranging species track their climate the worst, possibly because they're limited more by species interactions or habitat requirements, which I categorize as species interactions if they're biotic habitat types. But now, so stepping back from this work and looking at sort of the landscape of species range shifts and species interactions, I've been interested in how we can better attribute range shifts or variation in range shifts to species interactions and then push ourselves to be more predictive. And so I have sort of fallen into the same, I've been attracted by the kind of work that Laura and Gabrielle have just presented, looking at trying to combine a species interaction web with high resolution range shift data. And so for the high resolution range shift data, what we need is not relying on sort of global databases, but where my lab is going is to take our own data using environmental DNA, so we can sort of scale up fast observations of species distributions at large scales and do that quickly so we can do it through time. I wanted to say this, just that when we take range shifts and we try to ask to what extent human conservation efforts can help create more resilient communities in the face of range shifts, a lot of people are looking at what something like a marine protected area can do to maintain the resilience of a community. And there are lots of examples, as a matter of fact, where a marine protected area does protect the integrity of a community. And every time that happens, it's because of species interactions. So I just wanted to highlight that as like this connection between the importance of species interactions in range shifts and the role of protection conservation efforts. Okay, thank you very much, everyone.
Thanks, Jan. Sorry, no nepotism, so no questions for you. We can't. There's a few questions in the chat channel, so please go through them. I'm gonna give you a little break, Jan. So I'll introduce Ben, and then you take over. Okay. All right, so our next speaker is Ben Freeman from UBC, University of British Columbia, and he's going to be talking about latitudinal gradient in responsiveness of mountain taxa to climate change. Thanks, Ben.
All right, so this is very much following in the footsteps of what Jen was talking about. And I want to start by mentioning these studies that try to quantify extinction risk from climate change. It's getting warmer, species are on the move. People are very interested in the extinction risk, and some of the predictions are dire. In Nature in 2004, saying by 2050, 1/3 to 1/6 of all species committed to extinction. In 2015 in Science, more optimistic, but still 1 in 12 species predicted to become extinct from climate change alone. And so these are very scary predictions, and it behooves us to try to test to what degree do we believe them. And I'm going to focus on what's happening along mountain slopes, because there the models and the predictions are very clear, and the spatial scale is very short. So right now you have a mountain. It's warmer at the bottom, colder at the top. With climate change, every elevation is going to get warmer in the future. And the basic idea underlying the predictions is that species will track their climate. So a species that right now lives in the middle of the mountain in the future, in the warmer future, will live near the top. And a species that lives in the top of the mountain now will see its range shrink as it gets warmer. And then we should see some species that live only at the mountaintop run out of room. I and others have called this an escalator to extinction. Okay, so that's the basic logic underlying these predictions derived from models that assume temperature tracking. But a key step when we try to assess the degree to which we believe this is to validate the degree to which we believe our models. And the way that I and many others have gone around trying to validate is what Jen described, to look retrospectively. Climate change is not just something that will happen in the future. It's already been happening in the last half century or so. What have we observed happen? So should we, if we think that species will track their temperature in the future, have we seen them track their temperature in the last half century? If we think that mountaintop species are going to see their ranges shrink in the future, have we seen that already happen? If we think that mountaintop extinctions are going to happen, have we already seen these happen? And so those are the three predictions that I'm going to focus on today. And the second and third obviously follow from the first, that if species track temperatures, we will see these high elevation species shrink in range and mountaintop extinctions. So the idea that species track their temperature along mountain slopes. I've tested this by compiling resurveys that have measured the elevations where species lived in the past and where they live now. And so I have data, 146 temperature tracking scores for communities. And temperature tracking score is a metric that's the observed shift, how much we see the aggregate, the species in aggregate to have like one community shift is their observed shift divided by their expected shift given warming, measures of local warming. So if a temperature tracking score is 1, that's what the models typically assume, it means that their range shift will perfectly track changing temperatures. Species will track their isotherms as they move up mountains. A temperature tracking score of 0, on the other hand, means that in aggregate species in the community have not shifted upslope despite warming temperatures. And if it was negative, it would mean that species have actually moved downslope despite warming temperatures. And so here's the histogram of temperature tracking scores from this data set. It's the center of this distribution is clearly above 0. So in general, species and communities are moving upslope, but it's the center of that distribution is nowhere close to 1. So that the temperature tracking is nowhere close to the kind of perfect temperature tracking that is often assumed. So I call that fairly weak validation for this idea. Second, that high elevation species shrink in range size. Here, the data set is changes in range size for 975 species across the globe. And here what we did was we binned species into 3 categories, whether they lived at the top of the mountain, whether they lived in the mid-elevation, or whether they were lowland species. And to what degree did their elevational range size change between the historic survey and the modern survey. And so each black dot here is a species. There's tons of variation that we largely can't explain at this point, but there's no central tendency for mid-elevation or lowland species. Taken together, they do not have an overall change in elevational range size. Whereas for mountaintop species, we do see that your average mountaintop species is shrinking in range size. So yes, we do find some support for this, but with lots of variation. So third, has this set in motion an escalator to extinction? Are we seeing examples of mountaintop extinctions? And here, my reading of the literature is that there's really very few examples that we have. Pikas in the American West, Apollo butterflies in France, but given the amount of research interest in this topic, very few. So why is the validation for this very intuitive idea that species will track temperature along mountain slopes, why is this validation weak? And befitting the event today, I'm going to argue that the answer is that biogeography matters. In particular, the tropics are different. And we don't know the exact mechanisms, but it presumably relates to the fact that the temperature variation over annual and millennial timescales is much less in the tropics and much greater in the temperate zone. So now I'm going to show you that same temperature tracking data set again for communities. This time now I'm also showing you the map of where these resurveys come from. So there is more resurveys in Europe and North America, but there's some scattered around the globe. And I'm showing you temperature tracking as a function of absolute latitude, where we're called temperature tracking score of 1 means that the community is perfectly tracking temperature. And the predictions from a linear mixed model are that at the equator, the temperature tracking score is about 1. Whereas in the temperate zone, the temperature tracking is much less, meaning it's getting warmer in the temperate zone, but species and communities aren't moving up very much in the temperate zone. And that data is from a resurvey data set. We were worried that resurveys are pretty heterogeneous in taxa and methods and things like that. So we also looked at tree plot data set in the Americas, and we find the exact same pattern from tree plots. These are standardized analyses of the trees that grow in repeatedly surveyed plots. Okay, what about changes in elevational range size? I showed you these plots before. Now I'm going to break it down into temperate now, and tropical will come next. So in the temperate zone, like I already showed you, there's no central tendency for mid-elevation or lowland species to either expand or shrink in range. But in the temperate zone, the mountaintop species also have no central tendency to expand, to shrink in range. There's many high elevation mountaintop species in the temperate zone for which we have observed big range increases. In the tropics, there's a little bit less data, but still, especially for the mid-elevation species, some shrink, some expand. But nearly all the mountaintop species in the tropics are shrinking in their elevational range size. And so what I'm just showed you is that in general, tropical species are tracking their temperatures more. High elevation tropical species are shrinking in range. If so, we should be seeing more mountaintop extinctions in the tropics. And so to test this, I went to this mountain, the Cordillera Panteicoya in southeastern Peru, where in 1985, when I was 1 year old, John Fitzpatrick led a field museum expedition to survey the birds along this mountain slope and found these 13 species that only lived right at the ridge top on the Cordillera Panteicoya. This is an outline ridge that pokes up out of the Amazon. It does not connect to the much taller Andes that are nearby. And so I led a resurvey in 2017, and despite a lot of effort, we failed to find 5 species entirely. And when we modeled this using occupancy models, we have strong evidence that these species have actually disappeared rather than we simply failed to detect them given our survey effort. So what I want to leave you with here is this before we believe these dire projections, that trying to do our best to validate the degree to which our models are making good predictions is key. And I've shown you some evidence that these model projections work pretty well in the tropics, but not the temperate zone, and that this escalator to extinction is already in motion in the tropics. Already within my lifetime has led to the extinction of a high elevation community of birds in southeastern Peru, and we're actively looking to see if we have more case examples of this that have happened in the tropics, which we predict we will find. And with that, I'd like to thank the co-authors on these projects, and I'll either take any questions if there's time, or you can put them in chat. Thank you.
Thanks so much, Ben. Well, like everybody, I think we won't post questions through vocally, but we'll let you go through the questions on the chat. You've got a few. And with that, I would like to introduce our next speaker, Rachel Germain from the University of British Columbia, who will be, and Memorial University, who will No, incorrect. Definitely from the University of British Columbia only, who will be speaking about pattern process.
Oh, I'm so sorry.
It's okay.
She's going to talk to you about stuff. She'll introduce her talk, just in case I have the title wrong as well.
Great, well, I guess you can hear me. Welcome to my kitchen. There is somebody loudly making samosas behind me, so hopefully that's not too distracting. But I want to start with outlining what I think biogeography is. And from my perspective, it's the study of where species are geographically and why those species are where they are. And this talk very much is focused on the why aspect of this question and less about the where. And to answer the why question, I want to bring up this idea of the filtering metaphor, which we're all familiar with from ecology. You kind of see this in every introductory textbook. We teach it to our students often. And it's the idea that the species that we see in any one local community are the ones that kind of made it through this successive layers of filters and were able to actually persist somewhere on the landscape. And often what we end up finding or thinking that we find is that if we see this kind of trait distribution here, where we have these blue species that were able to pass through a blue environmental filter that are more similar to each other than some of the other species, but also more different in some other aspects. So even though they're all of a blue phenotype, it's really the most different ones that are able to actually persist together. That this is evidence of a strong environmental filter that has shaped that local community and allowed them to coexist with each other. But this is, of course, just a pattern. And so you notice probably in my talk title is that I want to get at process. And the idea here is that environmental filtering is not likely independent of competition. So it's not like the abiotic environment and competition are distinct filters. And as a little cartoon example of this, if we think about what environmental filtering is, sensu stricto, it's the idea that a species, if it were to try to arrive at some new site, so here a white abiotic environment, it would be unable to persist there regardless of whatever else is happening biotically. But the problem is that competitive exclusion can cause the exact same type of pattern. And so here we have a case where a species moved from a gray environment to a white environment, and in the absence of competition, it could actually persist there. But if competitors are present, in fact, it would be competitively excluded. And so you can see this is the same type of outcome that might lead you to infer that environmental filtering is happening. And the trick is that biotic interactions are happening everywhere, and so one of these scenarios is unrealistic biologically and one is realistic. The problem is that about 85% of studies often confound the environment with competition, or environmental filtering with competition when they're looking at patterns of trait distributions and things like that. So there's two questions I want to address in this talk. The first is evidence of environmental filtering consistent with the presence or absence of competitors? And does the ability, or does the answer to this question differ if we contrast among different abiotic environments? And so to answer this question, I first want to be clear in how filtering should be defined. And that's that these are being filtered. The idea is that if you were to add some species from that regional species pool into that local community, and they were unable to persist there, so their population growth rates were negative and they're on an extinction trajectory, that this is what filtering technically should be. And so the next question is, how do we measure persistence? And the way that I tend to do it is through an invasibility experiment. And I'm not going to go through the details of how to do this experimentally and also mathematically, but the basic idea is that if we want to test invasibility in the presence and absence of competitors, we must grow populations of species in the presence and absence of competitors and measure their population growth rates under those two conditions. And the idea is that we see where those population growth rates fall relative to a persistence threshold, so whether or not they're predicted to persist or fail. And if these population growth rates are less than 1, they're on an extinction trajectory. If they're greater than 1, then this is a case where these species should be able to persist in that community. And here I've replicated this kind of experiment in 2 different soil abiotic environments, a wet and a dry one, and I've done this for 30 pairs of species. And so what I'm going to start with is the first result, and take-home message of this is that environmental filtering only emerges with competition. To show you what I mean by that, again, we have these 2 soil moisture environments. This is the invasion growth rate that I mentioned. And first, we're going to look at these invasion growth rates in the absence of competitors. And this is all relative to this persistence threshold that I've outlined here. And there's going to be a lot of data here, but I'm going to walk you through exactly what you should notice. And the basic thing I want you to notice here is that all of these invasion growth rates lie above that persistence threshold, except for 1. And so there are very few species in this particular community that are being filtered in the absence of competitors. If we contrast this to what's happening with competitors, again, the thing to notice here is that persistence threshold. Now we have a lot of species that are falling below that threshold. And what's interesting about that is it means that environmental filtering isn't actually happening in the absence of competitors. You need competitors for it to happen. Another thing to note here is the slopes of these lines. Each pair of dots is a given pair of species. And so you can see how those species are responding differentially to the abiotic environment. In the absence of competitors, there's not a lot of slopes going on there. But in the presence of competitors, you now see that there is a lot of a response to that soil moisture environment. So the take-home message here is that competition is affecting the strength of abiotic filtering, and the abiotic environment is also simultaneously affecting the strength of competition. If we zoom in on a particular pair of species, in this case, a pair of congeners, Leucinia, this also makes a bit more sense. Here we have the invasion growth rate on the y-axis, again, against these two different environments. If we first look at those growth rates in the absence of competition, you can see that these two species are both doing very well. They're very high above that persistence threshold. They're not responding strongly to the environment, and they're responding very similarly also, and performing very similarly. In the presence of competitors, all of a sudden you see this kind of turnover where Glabrata does better in a wet environment, Californica does better in a dry environment. And in particular, if we look at where these pairs of species are falling along this persistence threshold, this is telling us that these species should coexist in a dry environment, whereas Glabrata should exclude Californica in a wet environment. Another aspect of this is this point that the environment can affect competition that we've, just like I've shown you, but it can actually do this in two distinct ways. And so the question is, which way is it doing it? To show you this in a cartoon, imagine the same pair of species that are competing in one type of environment versus another type of environment. There are two things that might give a species a competitive edge. The first is if one species just happens to do better in one of those environments. Forgetting competition, it just likes one environment over the other. Here you can see in environment 1, the species on the left does best. In environment 2, the species on the right does best. But the other way that you can have a competitive advantage is if species hurt each other more or less in different types of environments. So it's really on the per capita interaction strength of those competitors that is being affected by the abiotic environment. So again, if I throw this on here, what we're finding is that species on the left in environment 1 is competitively dominant to the other species, but this gets overturned in environment 2. This is interesting because when we're thinking about competitive dominance and how it's responding to the abiotic environment, people are often just focused on this first mechanism, and there aren't actually a lot of examples of how these per capita interaction coefficients actually change in response to different abiotic conditions. And so I'm going to show you a figure that's fairly complicated, but again, I'm going to walk you through the single few things that you should focus on. The first is that the x-axis here is something called a niche difference. It's basically a population pairwise competition parameter that is entirely driven by per capita interaction coefficients. And so the important thing here is if I have a species pair that are linked by that line, if it's completely vertical, this means that the abiotic environment is not modifying the abiotic environment, or sorry, the per capita coefficients are not being modified by the abiotic environment. But if there's any kind of slope to this relationship, it means that species are changing in their per capita interaction coefficients in response to the environment. Right. And so what did I find here? So you'll notice that there are actually a lot of pairs of species that are competing that do have this kind of horizontal structure to them. So there's many non-vertical lines. And this means that in fact, the abiotic environment is strongly altering for some species the per capita interaction coefficients, which again, this isn't something that we have a lot of evidence from in other studies so far. Another interesting aspect here is again, I've shown you how these different pairs are all kind of responding in different ways to these environments. But if you just kind of sum this and look at the distribution across those two different environments, we see that there's no difference on average between the environments. And this means that these species pairs are all kind of responding in idiosyncratic ways. Some are increasing in a wet environment and some are decreasing in a wet environment. So there's no kind of consistent environmental effect. And so to summarize everything that I've just shown you, the first is that environmental filtering only emerges in the presence of interspecific competitors. The second is that abiotic variation would allow species to coexist in some parts of the landscape and not others, challenging some models of, say, the realized versus fundamental niche. And the other is that the strength of per capita competitive interactions are idiosyncratic and shaped by the abiotic environment. And so there's this question posed in this tree paper from a few years ago about should the environmental filtering metaphor be abandoned? A response based on this data is that yes, most environments contain competitors, and competitors strongly modify persistence. But you could also say no, and maybe we're asking this question in the wrong way. Basically, what we've revealed are some interesting interplays between the species realized and fundamental niche through the biotic and abiotic component. And a plug for some of my other work, this is really focusing on competition versus the environment, but I've also shown that other filters of dispersal and environmental structure, the abiotic environment, are another aspect where it doesn't really make sense to look at filters as a hierarchical process. And so that's it. To acknowledge my co-authors on this paper, Margie Mayfield, and I think that's it. I have no idea how long that was.
Rachel, it was perfect. I was going to cough right when you said conclusion, so
right.
I'll let you go. Beautiful.
Yeah. If I can add one really quick thing about this is one thing I want to highlight, since we're all working remotely and a lot of our fieldwork has been impacted, it's that, I've been thinking about how we can continue doing research in our labs. And I think this paper is a really cool example because, it's an idea that I had and I really wanted to test. And I realized that I actually had some data already from another project that I could use to answer this question rather than doing something from scratch. And so I think, thinking about creative ways to use the data that we already have, given that a lot of our research has been impacted, I think is kind of a message I want to send to people. We're all doing synthesis science and stuff as well, but that's another aspect where we can still be productive.
Awesome. Totally agreed. And that's a great segue to our next speaker, Amanda B.
Jen,
oh, go ahead.
There is someone that has their mute off, and I've been hearing all kinds of conversations in the back. So if that person could please just mute, that would be great. Thank you.
Thanks, Pedro. Okay, everybody, check your mute. Thanks. Okay, and so if Amanda's here, she's going to be joining us. And Amanda is from Memorial University. She will be speaking about gaps in biogeographical data and future directions.
I have lost the share my screen button. Everyone from my lab right now who's listening is going to be laughing because I do this every day. So I don't see a share option anywhere. Am I missing it?
Can you sort of move, do you switch screens by moving over? Sometimes if I just click the Zoom icon on the bottom, I get a refreshed screen. Okay, she's doing it. Yay!
Yay me! Okay, everybody. So I am partially regarding the fact I didn't talk about science directly today, but it's my birthday. So when I saw that this was scheduled on my birthday, I thought, I'm just going to talk about something totally fun that I'm thinking about at the moment. So here we go. So I'll give a little shout out to my lab. We work on linking physiology to biodiversity patterns. I'm not going to talk about the science of that today. I'm going to talk about biodiversity data because this is when I think about biodiversity data a lot. How do we get it? How do we network across people to share their data? How do we work together? And how do we do that given most of us don't have a lot of time? So it's great to think about all the things we want to do, but there's some challenges there with time. Okay, so this is a vision that I share with Maria Dornelis and many other colleagues. And the idea, and this has been put forward by many, many other people, but I love what Maria did with this particular paper. And it's thinking about a macroscope, a way of observing our Earth through something like a microscope. But now we need a macroscope because, of course, the Earth is quite big. So if we think about how we want to do that, that's going to involve a huge network of instruments, ways of observing nature, pipelining that data, streamlining that data, and integrating it so that it can then be used. And I am certain that all of us today have questions that they would love to ask, but they just don't have the information to do so.
Amanda,
Yeah.
I'm going to just interrupt. You're on, your screen share is showing your presenter view. And do you think that's a cool?
You know what I'll do? Maybe it's this, Jen. Is that better? No, it's it.
It's better. There, perfect. Go there, do that. Okay.
Thank you. Okay, so I've been working with colleagues. This was initiated by Anne McGurran and Maria Dornelis, and there's a large core team of us now that are aiming to collect data from around the world and combine this into biodiversity data and disseminate it and create it so that it's open. So I'm going to talk in numbers that are likely correct to an order of magnitude. So we have about 1,000 studies, and in those studies we have, say, 1 million rows of data. And what's really apparent is that we have gaps in taxa and space. And Laura Pollock did a beautiful job of talking about these gaps already. And I think what's really key to think about is we've got these gaps in taxa, we've got these gaps in space, we have gaps in time. So how do we start to fill these gaps? And what's interesting is when we think about these gaps, what it means is that when we try and understand how biodiversity is changing on Earth, underpinned by species distributions and other shifts, it means that we're fundamentally limited in how we go about doing that because we don't have all the information. And the information we do have often has inherent biases and sources of error which are difficult to account for or can create kind of, I guess, systematically bias how we understand how the world is shifting. And this has created a lot of controversy as we start to dig through these first initial data sets, looking at how entire intact communities are changing through time. And it also biases our assessments, and even in doing so, how we engage our public and how we talk to public about how biodiversity is changing. And because of these messages, it might even feed back into society so that we write books about animals that are charismatic, and we miss sharing at early stages stories and information about other amazing taxa or systems on our planet. And so we're really lucky in Canada. We have the Geobond Secretariat, Andy Gonzalez, who's now at McGill University. So we have this incredible opportunity to work together as a team across freshwater, marine, and terrestrial systems to think about how we can work together as a group to kind of observe our ocean systems and hopefully create some really great sets of data that we can then ask some really cool questions. And what really strikes me in coming back to Canada is there's some amazing data sets in Canada that date back both in space and time. And so I'm really hoping that we'll be able to kind of network and include a lot of those data sets. And as we move from thinking about biodiversity data and we think about, well, let's try and understand and identify some possible mechanisms which might be driving change on our planet. And we look at the data that are available on a global scale to understand what is changing and where. It's also immediately obvious that a lot of that data is also fundamentally limited. So we can certainly create layers on climate change or human use, human population, pollution, and invasions. And we did this for the whole globe and merged these layers across marine and freshwater systems. But even in the review stage, there were a lot of criticisms because the fundamental layers that were feeding in to these, I guess, environmental layers were limited themselves. So I guess a real challenge that I think we face launching into this era where we're having more and more data that are available, greater opportunities for networking across fields, across disciplines, and in kind of teams of scientists, is that we can now start asking for the types of data that we want. And one interesting kind of aspect is that when we needed to predict weather, it meant that physical kind of environmental scientists then grouped together to create standards and protocols for submitting information so that that information could then be compiled into larger kind of streams, and then from there collate information so it could then be disseminated. And we, as ecologists, we use so much of this satellite and other information in terms of the work we do. but this information isn't necessarily relevant to the individuals and what's actually impacting them as they go about their day-to-day lives. So we're increasingly working together in large teams to increase these kind of gaps in the environmental information we have. And it takes a lot of time, it takes relationships. So I'm hoping we're going to fill the gaps, and a lot of what we've heard, a lot of what we hear about are forests and plants and other systems which we can access. But if we think about these systems, they've already been fundamentally impacted by humans. And there's other systems on the earth that it might make sense to start testing these fundamental ecological hypotheses like deep sea or more remote systems, because they're relatively less impacted by humans. But maybe not for long. I'll talk about that in a bit. So I think when we do this, surprises will start to emerge. And what happened when scientists put out temperature loggers at deep sea hydrothermal vents and started collecting temperatures is there was this is at 2 to 3 kilometers in depth on the sea floor, is the temperatures of those hydrothermal fluids varied with storm tracks as well as with kind of lunar cycles. So the moon and storms are affecting the temperatures of hydrothermal vents, sometimes by many degrees. It's quite remarkable. And of course, what we're seeing right now in terms of the pandemic, which has been going on for thousands of years, is that human beings are intimately connected with nature. We're a part of this. And we've already fundamentally altered kind of the planet. And what I decided to do with a couple of co-leads, Richard Primick from Boston University and Carlos Duarte from KAUST University, is to start to collect all of the information that we could on how human beings relate to biodiversity on Earth. And there's some fascinating kind of take-home lessons from this. And I'm not going to go over these today. But what I'd like to emphasize is that when we think about our lockdown, and we've confined humans, that's affected all sorts of societal systems. And in turn, that has had these kind of linkage effects to biodiversity threats, pollution, exploitation, for example. And then in green, a number of biodiversity responses, movement, survival, abundance. And those in turn are affecting the very ways in which we go about protecting nature or collecting data. And in particular, our environmental monitoring and science, and probably funding for science is going to be hard hit. So we're all kind of connected in this real connected network, but it's very clear that human beings have re-engineered our planet, and we're moving to the very deepest sections of our ocean now to mine basically minerals such as near hydrothermal vents, but in sulfide areas, and mine those minerals for things like cell phones. So this is happening, and we're going to fundamentally alter our deep systems as we have much of the surface of the planet. And this, I think, is really key to think about when we think about biogeography, because we're dealing with a very re-engineered planet. And so when we ask where are species in time and space and why, if we're going to move forward, I heard the cough, Jen, if we're going to move forward in time and get all of this data together, how do we do this? So I'd like to end with just 4 kind of suggestions. How can we do science differently? I really think we need to share our vision and we need to share our data. We need to start filling those gaps. But I still think there's something that's a little lacking, or perhaps something that's blocking us somehow. I'm not sure how to get on with this. But in thinking about the fact that this was kind of Humboldt Day and what an amazing accomplishment we've had for biogeography, I also started to think about the fact that how we actually do our science and even the questions we ask are rooted in kind of a colonialist background. And I think as we move forward, when we start thinking about how do we embrace this kind of connected and data-rich world, that we might learn by embracing diversity. So my last slide is just an example that I found online that showed something that I probably wouldn't have seen 15 years ago, which is a field school from UVic teaching about ecology, economy, and spirituality from a Peruvian teacher in kind of a field setting. I think this is pretty interesting, and I think as we move forward, thinking about diversity in biogeography would be a great goal. And so with that, I think I'll end and promote beautiful Newfoundland and the beautiful institute where I work that's situated right on this gorgeous cliff. Thank you.
Thanks, Amanda. Happy birthday, Amanda. Well, I'll let you check the chat for questions and move to our next speaker, Jean-Philippe Liza from Concordia University, who will be talking about biogeography of host-parasite interactions.
Hi, Jennifer. Thanks. You guys, are you seeing this correctly?
Yep. Yes.
All right. Hi, everyone. Very impressive sets of talks today. Very interesting also. And I think the talk I'm about to give is very much related to what Kay Jones talked about earlier today. It's about parasite geography, and it is something that we are just starting to explore in the lab right now. And more broadly, we're interested in biotic interactions and how they vary in time and space, and how does that affect species distributions. Some of the reasons to care about this are that, well, there are implications for ecological theory, but also the ability to predict where species will be in the future and how they'll interact in the future can be very handy when it comes to disease ecology, disease spread. Where will disease be in the future? Where are we more likely to see them? It is also interesting in the context of climate change, because as we know, biotic interactions can mediate where species are now, but they can also mediate where species will be in the future. And that's an example here of a paper by Leslie Lancaster, where she shows that host herbivore interaction would mediate the movement of butterflies during climate change mediated migration. And when it comes down to host parasite interactions more specifically, it is quite challenging to even understand where are you more likely to see a high prevalence of parasites and a greater diversity of parasites. Some research is showing that it often requires knowledge about the host community interactions, and so how transmission can happen within the community, as well as information on the underlying variation in the abiotic environment, and finally information on host characteristics. And so in the lab, we've increasingly studying dragonflies and damselflies, which we refer to as odonate, which are sometimes heavily parasitized by aquatic water mites that attach during the larval stage in the water and just follow the individual dragonfly throughout development all the way up to the adult stage. And as you can see here, this parasitism by aquatic water mite, mostly the genus Erinerus, can become very, very intense and have fitness consequence on the individual hosts, which can potentially trickle all the way to affecting populations. And that is why understanding the geography of these parasites and the intensity at which they infect their hosts can have potentially important implications for population dynamics and species distributions. And so as we were studying odonates, one undergraduate student in the lab, Daniela Lacerbo, who led that study, started counting the mites that we found on our odonates along a gradient reaching from southern Quebec to northern Quebec. And she found that parasite intensity, right, which is the number of mites that an individual has on itself, along with other metric of parasitism, varied systematically from south to north, and that in temperate regions, on average, you tended to have quite higher rates of parasitism and more intense parasitism. And so why is that? And that's what's driving this research, is we're trying to understand what's driving this geographic pattern of parasitism. And there could be a suite of potential variables that do matter. And here we're just going to classify them into two big categories for simplicity. Abiotic variables that relate to the environment, which could have direct effect on populations of the parasites, and biotic factors, which relate more to the host community and host individual themselves. So we were set to try and see what's the role of these two sets of variables for influencing the distribution of parasitism. This is a 36 lakes that we sampled from southern to northern Quebec. And then at each lake we had a standard protocol for sampling. So that allowed us to have a kind of a same basis to compare relative abundance and composition of our communities. And we, while we measure several abiotic variables, which I'll show in a second, but we also collected in total 843 host odonate across 42 species. And we counted over 2,000 parasitic mites. Some individuals have no mite whatsoever. Some species have no mites whatsoever. And other species and individuals can have up to 100 mites on them. So we built a Bayesian model to try and assess the influence of variety of variables. Water chemistry is one of them. We know that water pH affect the composition of odonates, hosts, and those lake. We look at things like size of the lake, dominant vegetation, and climate, which have been shown also to have an influence on the host and could also have an influence on the populations of the parasite. In terms of biotic variables, we look at the body weight of the host species, the relative abundance of the host species in the local community, and the phylogenetic structure of the host community, which sometimes can relate to transmission, right? A lot of closely related hosts in one place might favor transmission and yield a higher average intensity of parasitism. We also had a site random effect and a species random effect in that Bayesian model. So because we had an aspect of community phylogenetics, we used this tree, which we developed in the previous study published in Aerosmith et al. In 2016. So that's the molecular phylogeny that we use for that part of the analysis. Here you're seeing the coefficient, the standardized effect sizes of the different effect that we influence, and so that we assess, sorry. And so what you can see is there's a very strong species effect, and there's also a site effect. And then if you try and look into what are these species and site effect in more detail, there's a suggestion that first of all, the species effect is a phylogenetically conserved effect. So more closely related species tend to have more similar level of parasite intensity. And it turns out that this phylogenetic effect is driven by body size. So closely related species of hosts have similar body size and are more likely to have high parasite intensity, and this relationship is negative. So if you have a small body size, you're more likely to have a high number of parasites on you. And on top of that, we had a side effect, and that side effect turned out to be mostly one that relates to water chemistry, and in particular pH. And our analysis suggests that pH effect is not mediated by the impact on the host communities, but really directly on the parasite populations. So looking at this result in more details, this biotic factor, this body size effect in particular, this is the phylogeny of the species recorded across our 36 sites. And on the left-hand side there, you can see in blue the number of mites per individual. So each dot is an individual odonate. And you can see there's a lot or a few parasites on that particular host individual. And on the right, you can see the corresponding body mass for these individuals. And so this cool pattern emerged that I was mentioning before, where smaller species as well as smaller individuals within species tending to have more parasites, and that effect is phylogenetically conserved. So it is tempting to say that there is something between body size and immune response or immune protection against those parasites. And in fact, we were finding this idea that large and dark odonates tend to have few parasites, and small and lightly colored odonates tend to have more parasite. There seems to be a correlation there, and some of these patterns have been documented before. So Ilvonen and Suhonen showed that if you just look at a comparative study of species in Europe, you do find that smaller odonates will have a higher prevalence of mites on them And they also find that it is conserved in the phylogeny, and they also find that the immune response is correlated with all of this. So if you're small, light colored, you tend to have weaker immune response against the mite. And so it might be that body size itself is not really what protects the dragonfly against the mite. Could be that body size relates to the strength of the immune response. Body size correlates maybe with color, which has to do with melanization, which is how the immune response proceeds. Or it could also be that bigger dragonflies have thicker cuticle, which protects them against the parasite. So we don't know, but there's some phylogenetically correlated trait that seems to have an impact on how susceptible you are to parasitism. And now we are pushing this research further with undergraduate student Maggie Blondeau, who I think is listening right now. And Maggie has spent the last 2 summers sampling odonates and performing thousands of immune response measurements in an attempt to understand the geography of immune response, which we think will illuminate our understanding of what drives geographic variation in parasitism among individual odonates. So in conclusion, I wouldn't claim that odonate-mite interactions will allow us to understand disease spread, and predicting it in humans, because it's very unlikely anyone will be infected by a mite, a parasitic mite, or have a disease transmitted to them by one of those water mites. But it is a good system. It is to study these types of interactions, which may be, generating knowledge that can be transferable to other systems in the future. And clearly, our results suggest that if one wants to predict where in space you're more likely to find a high number of parasites, well, you need to know something about the relation between that parasite and its biotic environment, but also you need a lot of information on the physiology of the host and the susceptibility of the host to the parasite. And it's really the interaction of these two things that will help us better predict where high level of parasitism might be encountered. And I mean, there's a lot of implication or applications for the sorts of research. I think we're seeing a lot of dragonflies, butterflies, lots of insects responding to climate change and moving up north in Canada. And it might be interesting to think of, are they going to bring with them more disease, more parasites as well with them? And these sorts of models we're developing, which are understanding the processes, might then be used perhaps eventually to create predictive models that will help us address some of these important environmental issues we're facing right now. So thank you for your attention, and if there's time, I can take questions.
Thanks, JP. That was great. Sorry, Jen, were you saying something?
Nope, I was just going to pass it over to you.
Okay, sorry. Thanks, JP. I think we'll do with the others. Maybe take questions over the chat. I'll save the chat later so that I can maybe send questions that were not answered. I would like, before we go to our live discussion activity, I'd like to invite and ask George Stevens, who is a board member of the International Biogeography Society, and to ask him if he could say a few words on behalf of the society. George?
Thank you very much, Pedro. My name is George Stevens. I'm the treasurer of IBS, serve on the board. This Humboldt Day series of lectures is really quite impressive. We'll be working all week. Please go to the humboldtday.org website to see what other speakers are coming up and what other locations. But as Pedro pointed out early on, we literally have hundreds of people participating today, and as we go through the week, we will be in dozens of different locations. This is a really challenging time for IBS. Our essential function is to host face-to-face meetings, to try and encourage collaboration between scientists, and to encourage those conversations that you can only really have over a beer. What's really great about this video conferencing system is, of course, the chat, and you can contact people and talk. But the challenge for us on the board is to try and figure out, when do we do our next face-to-face meeting? We have been on hold. We are interested in technological change, but we're also interested in your opinions on how we can foster the collaboration that, as I said, only comes from face-to-face conversations at a meeting. So I'm most interested in comments you can add to the chat, either now during the discussion or later on. We will look at all those comments. But the question for the board is, first of all, do these sessions help you? Do you enjoy them? Are there things that we could do? And secondly, how can we creatively address the face-to-face meeting issue? What are ways that we can think about how we can use either video conferencing or other technological tools to create the same kind of atmosphere that we all enjoy at face-to-face IBS meetings? So I just want to end with thank you to everyone for participating today. Your involvement in IBS is what makes it great. And these Humboldt days will be made better by your participation. So thank you for that. Pedro, I'll turn it back to you.
Thanks, George. So this has been great. I would like to thank the IBS, the International Biogeography Society. It's been fun to have, to see people again. Hi, Richard. In particular, to thank Karen Faller, Miguel Matias, and Sandra Noguet, who did a lot of the background work. And for the Canadian event, Amanda Bates, Jen Sunday, and Jonathan Davis were more of my partners in crime here. So I'd like to thank everyone. I'd like to thank you for having come. Thank all the speakers that came and provided much food for thought. It's just Monday, so again, please visit the IBS site. You're going to find lots of interesting things there. Jen, you want to say something? Amanda or someone else? Anyone is welcome to say something.
Hey, you guys, thank you. I really enjoyed this. I think I'm very proud of our Canadian event, but I will go and check out the other events online. And Pedro really was the cheerleader for this project and did the lion's share of the work. So thank you, Pedro.
Thanks, everyone. I'm going to go prepare my lectures for the week. All right, everyone. Good seeing you.